kind of an interesting proposition. That is the idea that human life can be extended almost indefinitely. We have the technology in hand right now, theoretically, to do this. What, of course, are lacking are laboratory facilities, medical facilities, and the will to do this. A lot of political issues are involved here. Could we have that first slide? Many of you are familiar with the writings of Robert Heinlein, the science fiction writer, and the adventures of Lazarus Long. Lazarus Long, in his most recent incarnation in our imagination, is achieving his enormous lifespan through the use of telomerase. Telomerase is an enzyme which has been discovered which extends the DNA molecule once it's been ravaged by replication and renews it so that it allows the cell to continue dividing indefinitely. The subjects which we're going to cover this afternoon are non-dying germline protoplasm, perils of Earth habitat, medical hazards of astronautics, which we'll touch on later, longevity for interstellar spaceflight, youth extending telomerase, cryopreservation of useful immune system cells, longevity research on the International Space Station, culturing and freezing of the astronauts' tissues, and finally prospects for indefinite extension of youth. If that sounds interesting.
Here's a picture of Here's a picture of Madame Jean Carbon of France. She died in 97 at the age of 122 and 5 months, achieving the longest documented life in human history. She was a French radio program anchorwoman and had a very varied life. She sold paints to Van Gogh later in her youth. She had made 122 trips around the sun, but her protoplasm had been alive a lot longer than that. As a matter of fact, it had been alive as a living entity for 3.8 billion years on this planet, which is the current estimate of when life began on this planet. And a round trip around the galactic center is sometimes called a cosmic year, and it takes 200 million years to go around the galaxy once. So 3.8 billion years of Jean Carbon's protoplasm made 19 cosmic years long. That's a long time to be alive. We're not saying that the protoplasm is immortal, that it couldn't be killed, but it certainly is youthful. It doesn't experience a thing called biosenescence, the gradual loss of essential function, which does in most of the somatic cells of all multicellular organisms, including ourselves. Life, for quite a while, however, maintains itself in a condition called homeostasis, where 5 or 6 different juvenescent processes try to reestablish the equilibrium without you having to worry about it. You can sleep, you can do whatever you want. Your body is self-repairing, removing defective cells, regenerating new cells to replace them from stem cells, recalibrating your thresholds of endocrine and the nervous system, and also incorporating better genetic sequences from the environment. So we call that the 5 R's of luminescence. And there's a subset of cells in nature of all the species which doesn't age. It's called the germline of cells. We have such cells also, as you know. We have children, and our gametes are unsenesced. The reason why the germline cells don't senesce and the somatic cells do were unknown 40, 50 years ago, when first we wrote a memo in the Air Force to try to find out what kind of a longevity astronauts would have to get to the nearest star system to look around for planets and so forth. And I was asked to write a memo to the general in charge on a speculative trip like that, we assumed that 10% of the speed of light as the coasting speed and came up with a 100-year round trip for the astronauts, obviously not long enough for current astronauts. I would like to digress for just a minute to sort of remind you where we are in the space program right now, namely two events stares in the face. One of them was the dumping of the ashes of the astrogeologist Eugene Shoemaker, the discoverer of the comet Shoemaker-Levy 9, which impacted the planet Jupiter in 1994, and killed himself in Australia 3 years later in an automobile accident. Dr. Porco of our Planetary Sciences Department at the U of A was instrumental in getting Shoemaker's ashes dumped on the lunar South Pole crater Morse last July 31st. These are the first human remains buried anywhere outside in outer space. And at that same lunar South Pole, the next slide perhaps. This is a picture of the several segments of the comet Shoemaker-Levy I was just talking about, impacting the planet Jupiter. And from left to right is segments A, C, D, F, E, D, G. Would you turn that slide around for a minute, upside down? It seems like the planet wasn't all that happy about it. Next slide, please. This is the south pole of the moon, and in the center of that picture taken by the Clementine probe is the crater Morse, where Shoemaker ashes are buried. Nearby is the crater Shackleton, on the rim of which there's a thing called the Peak of Eternal Light, sticking out, enjoying 24-hour sunshine. The German and European Space Agency is fixing to put a generating station right there. And then 120 kilometers away on the Mount Malapert, they will install a transmission station to the Earth so they have continuous communication with the moon and don't need any batteries because they're getting free solar power. So while we make a burial site there, the Europeans are making a first installation. On the other end of the spectrum, could I have a few more slides here, please? This is where the ashes are buried. This is where the arrow points is where the first human ashes are now on the moon. Next slide, please. Here I've circled the crater Morse, which is the burial site of the ashes. They were looking for water in that deep crater, thinking that they might find it there because of the shadow. Next slide, please. This is just a report on what I just talked about, the Europeans establishing these two stations on the South Pole. Please, next slide. This bird is called the Titan 3C. It was our first heavy lift vehicle. It was first launched in June of '65 with a payload to go into geosynchronous orbit, communication satellites at 22,000 miles above the Earth, which rotate around the Earth in 24 hours, so that's called geosynchronous. And you notice that this particular vehicle has a trident shape with two strap-on 6-segmented solid rocket motors, and that same vehicle with some upgrades is now still the so-called free world's or non-communist world's most powerful rocket. Next slide, please. That's what it looks like now. It's the Titan 4B Centaur configuration, used for military payloads as well as deep space probes. Next slide, please. There she goes to the planet Saturn, launched October 15th, 97, arriving there November 6th, 2004, with a Cassini probe, which has in it the Huygens probe, which it will descend on the Saturnian moon Titan 3 weeks later by parachute. Next slide, please. Here you see the Cassini orbiter, having just separated the Huygens probe. Underneath is the surface of the moon Titan, Titan being larger than our planet Mercury, but still a moon because it's around Saturn orbit. In the distance, you see the planet Saturn. Next slide, please. Now, the Voyager passed by only showed a hazy orange surface because they couldn't penetrate the organic smog in the atmosphere of Titan, which is mostly a nitrogen atmosphere. Just the only other celestial body in the solar system with a heavy nitrogen atmosphere like the Earth. However, next slide, please. The Hubble telescope, using infrared photography, could see through that smog, and we see certain surface features still very vague, but you see a whitish area, which is the continent the size of Australia there above the methane ocean. Next slide, please. Now, this was taken by the Keck telescope on top of Mauna Kea in Hawaii. The experiment run by people from Lawrence Livermore Laboratory in Berkeley, and they use speckle interferometry, which allows them to eliminate the Earth atmospheric haze and produce the sharpest picture yet of the surface of Titan. You can see a rubber duck-shaped structure there, which is thought to be water ice. So what we have on that planet is water, oxygen, nitrogen galore, and many organic molecules, everything needed for life, but it's very cold, minus 280 degrees Fahrenheit. Next slide, please. There's a rubber duck enlarged. Next slide, please. Well, here you see the Huygens probe parachuting down on the surface of Titan, the farthest point ever reached and touched by any man-made object in history of space program. The date, November 27, year 2004. Next slide, please. Now, having covered this, I'm returning to the main topic, which is that we are talking about extending the longevity for astronauts to be able to make more distant voyages. The current lifespan not being enough for proper exploration, and we feel that the survival of our species requires such extension because we never know when we're going to get hit by something like the comet Shoemaker-Levy 9, which would really be a problem. So the search for new habitats is essential for ultimate survival.
It's clear that from the foregoing that the Earth is in trouble and we need to do something about it, like get out of here. And there will be essentially there will be a detailed discussion of this in a session tomorrow about the perils and so forth. We've listed here a translation of NASA's list of perils, and I think it's evident to anybody who's considered the problem that we need to take several approaches. Our approach essentially is one in which we are going to extend human life and to enable us to undergo long space trips. Just simply getting out to the outer planets will not be sufficient. When the sun expands as a red giant, it may make the entire solar system uninhabitable. And thus we'll have to look towards extended interstellar travel. And there are several ways of achieving that, but one of them is to keep the astronauts who are undertaking these voyages alive indefinitely. As improbable as this seems to people who have not considered the question, it is a possibility, and my colleague Chris is going to tell you a little bit more about how we're going to work that problem.
We have, unlike what Ponce de Leon thought, an internal fountain of youth. He was looking in Florida, at Bimini or someplace outside. The fountain of youth was inside of him all the time. The fountain of youth is the early stem cells of our body that haven't aged and differentiated very much and can be used to constantly rejuvenate your body. And you can culture different cell groups from them. And so if you tap that internal fountain of youth, you can do dramatic life extensions in theory as well as in practice. As I mentioned earlier, when I described this memo, I had to write to the Air Force as a captain then, to a general who wanted to know what would be involved in such interstellar travel. Could I have that memo, the next slide? This is written in June of 1959, before the first manned space flight, and it was addressed to the Deputy Commander for Space of the Air Force Ballistic Missile Division, the outfit that built all these big rockets in the Cold War. And towards the word interstellar appears in the upper paragraph. Lower down, there is a question raised of why the so-called germline cells are immortal, and most of the somatic cells are mortal and age and die, and there should be a biochemical difference discernible for the two classes of cells, and that the Air Force should investigate that. As you can see, that was raised in connection with the early space program in an official document, and NASA picked that up also at the time. However, the answer to that question had to wait 25 years until Carol Ryder, now at the University of Johns Hopkins University, discovered the enzyme telomerase, which is a reverse transcriptase enzyme that re-adds the snipped off ends of chromosomes, the telomeres, and keeps them always healthy. However, in the absence of that enzyme working, each time a cell divides, a piece of the end of the chromosome gets snipped off and not replaced. After about 50 such divisions, it reaches a critical thing called the Hayflick limit, and the cell quits dividing, lest it be actually cutting off important genes. So the cell becomes post-mitotic, and as it doesn't divide anymore, wastes accumulate inside the cell, squeezing the microtubules out of the way, and the cell starts to senesce. There goes your aging process right there. In certain cell groups, such as eggs and spermatozoa, as well as cancer cells, the gene that templates this enzyme is very active. So there's plenty of telomerase in spermatozoa, eggs, cancer cells, and also some cells of the intestine. Research is now underway at Geron, Incorporated in California, to try to add telomerase to somatic cells to get them to become more useful and reverse the senescence by that administration. This is also being picked up by the National Institute of Aging, who are now funding by millions of dollars precisely that type of research. Next slide, please. Oh, this slide shows how farther out voyages, many astronomical units away, require increasingly amount of an extended life, and this is a logarithmic scale. So as you look at it, the life increments are really much larger, but it's a logarithmic scale. Next slide, please. This is just a printout of the National Institute of Aging having established special centers at 5 universities, each getting $7 million to study the aging at the gene or genome level. Next slide, please. By the way, how about I think, Kent, you have something here to talk about. I was just going to mention that there's a concept that was first brought forth by a researcher by the name of Takashi Makinodan of the NIH called the immunological rejuvenation. He coined that phrase. We are doing that. Our company is the first company approved by the FDA for doing things like that. Ken, you want to talk about that a bit?
Yes, can we have the next slide, please? Our company, the Ageria Group, has, over the last 10 years, has investigated the possibility of preserving the immune system, the human immune system. As we know, the immune system deteriorates along with the rest of us during our lifetime. And this makes us much more vulnerable to the disease processes in our 70s and 80s. What we do is extract blood samples from people in the prime of life and centrifuge out the white blood cells and preserve these in liquid nitrogen. And then when you, in your 80th or your 8th decade, we can reinfuse these, and they act very much like bone marrow transplants. They can resuscitate the immune system without all of the fuss and bother, and they are perfectly histocompatible. Whereas bone marrow samples, as you know, are very difficult to find and are not histocompatible. Anyhow, the Immunobank program has had some volunteers. Several people here at the conference have actually had their stores cells stored, and we are planning to do a new series of tests to determine the efficacy of this process. It's been approved by the Federal Food and Drug Administration, and we anticipate that we will be able to begin this process on a commercial level very shortly. Can I have the next slide, please? We are also conducting some other aspects of this in regard to the genetic aspects of aging, and my colleague Chris will discuss how we're doing this genetically.
What you're looking at is a slide in a microarray scanning machine here at the Arizona Cancer Center. Many medical centers now have these. It is possible to examine as many as 5,700 genes in a single slide by dyeing the samples and the probes with fluorescent dyes, which then illuminate the genes in question. And they respond differently to the dyes depending on how active the gene may be. If it's constantly transcribing RNA or so forth, it might fluoresce in one color, whereas if it's just doing it a little bit, it'll be a second color. If it's been down-regulated, it'll shut off altogether, it'll fluoresce in yet a different dye. This can then be measured, and by a computer you can find out which genes are up or down-regulated. It's a new technology. We have frozen cells that have been frozen solid for 8 years already from our volunteers, and the same volunteers are now volunteering to give new samples, and we'll be able to compare what happened in the last 8 years to their genomes, their gene banks, in their cells to see which ones have shut down, which ones have been reactivated incorrectly, and so forth, and actually track the aging process at the gene level. Another question that might come up in this connection is whether we can use stem cells to grow our own body parts. This is called nuclear transfer technology, as you know, all know about Dolly the sheep in Scotland and so forth. By the way, while being an genetically identical twin of her mother, though was already aged because the DNA used for cloning Dolly was an aged kind of DNA. She was born aged. Jaron is trying to rejuvenate her with adding telomerase artificially, and so is the University of Texas Medical Center doing this kind of work. Am I still on? If so,
Discuss the blastocyst.
okay. The way nuclear transfer fertilization is done is you take a diploid nucleus from either a woman or a man and inject that nucleus into an enucleated human egg, which has no nucleus at all. Therefore, all the DNA in this fertilized egg is from just one person. This will be a genetically identical twin. The first 3 divisions produce totally potential cells. Any one of the 8 cells could make a complete embryo. After that, the next 3 divisions end up in a blastocyst. There's a picture of a blastocyst. It's a 64-cell structure which has a cavity in the center, a coelom, and on the side there, perhaps you can read it if not, it's called the inner cell mass. It contains all the precursor cells of your 250 different kinds of body somatic cells. You can harvest that inner cell mass, and there's no connection between that and growing your own twin. The harvested cells simply can be used to grow your spare organs, spare tissues, any cell type you wish. 2 weeks in culture, this bunch of cells taken from the inner cell mass will develop myocardicytes that can be seen beating already 2 weeks later. So it's a terrific source of replacement fresh stem cells, totally, of course, compatible with you, your own cells. So we think that this is a very important thing of future life extension, because we'll be able to replace aging tissues left and right by this technology and maintain what's called the atarasia, which is in medical dictionaries, if you want to look it up, meaning the extended youth of an adult person. And so the medicine, the branch of medicine that would rejuvenate you, we call agerastics, after the word Algeria, and I think Ken has something more to say about it.
Could we have the next slide, please? These curves here on this graph, many of you don't much care for graphs, but we're attempting to illustrate here what happens in the aging process. At the bottom of the scale, we see age in years, and the vertical scale represents human capacity. The normal aging curve, which we call the Ugaric standard, usually ends somewhere in the 8th decade. Actually, that's optimistically speaking. And it's pretty much what we all expect to do on this earth. This really means that we have not been engineered, and that, in fact, we're not even optimized. We're subject to all of those problems of aging. There's another curve alternative, which represents the lifespan of someone like Jean Calment, who was genetically inherited longevity from her parents. But we believe that that third curve up there says an engineered asymptotic agerasia. And what this really means is that ultimately we will be able to engineer the human somatic cells and to an indefinite lifespan. It doesn't mean, of course, that you will survive somebody hitting you with a truck, but what it means is that your cells will continue to function as if they were optimized in some stage of youth. And so with that, we know that we have the theoretical evidence for it. What we lack are some of the laboratory techniques, but these are being developed in a very explosive fashion. With that, I'm going to close and take some questions from the floor on this process.
Can we have the next slide, please?
Yeah, that's
We
right.
just want to remind you what the biggest, strongest rocket is in our space program. There it sits on pad 40 at Cape Canaveral, is Spaceport, USA.
Thanks. Yes.
Hi, thank you for a very interesting presentation. We're sitting here noodling over the very nice hypothesis study and exercise as well. But a couple questions. One, you were talking about freezing cells.
Yes.
Certainly we have evidence that if you cool cells or cool an organ system that you can also extend longevity. I mean, there's resuscitation work that shows that. And I'm curious if there's any consideration to using just cooling, or taking several different cell lines, cooling them, let's say, from body temperature down to 35 degrees Celsius, down to 32, 30, and so forth. We may be able to find that there is a longevity curve And instead of freezing cells, maybe we can just cool them instead. And is that going to be easier in terms of storage or whatever?
Well, sorry, there's certainly, you're correct in such an assumption. Hibernation and reduced metabolism has been explored for many, many years, and there certainly is credibility to all of those approaches. But I think when you're talking about absolutely indefinite span, you have to stop the aging process. When you cool people down, you slow it, but you do not stop it.
But maybe you don't need to stop it.
Well, perhaps not. In fact, in near-in solar system flights, it may be sufficient to put them in hibernation, as was certainly explored in 2001 and so forth. But
I'd like to add
for 30 lifetimes, you need really to consider stopping the aging process.
The experiments for the last 20 years on Space Lab, Mir, shuttle and so forth, that examined the effects of microgravity free fall on cells, especially immune system T cells, shows that the cells, no longer being in compressive gravitation, quit going through the cell cycle and don't replace themselves. The same thing is happening to microskeleton structures in cells. In other words, the lack of compressive gravitation and opposed gravitation is causing these cells to no longer function. So astronauts returning from extended space flights have a downregulated immune system. We are asking NASA to check whether if they were frozen solid, like our immunobank program, then they would not be susceptible to the effects of that microgravity.
Yeah. Another question?
Yeah, but the piece that still remains, the variable that's controlled for is the lack of gravity. Just because you may be able to arrest the aging process doesn't necessarily mean that the effects of lack of gravity can attenuate as well. So that's a question that has to be answered.
Well, we're assuming that these are microchemical processes which come to a halt when you subject the living organism to temperatures of liquid nitrogen. Virtually all chemical activity at the molecular level certainly ceases, and the expression, the misexpression of the DNA molecule under microgravity conditions is really at the root of these problems about impaired immune system and so forth. I agree that the subject is not thoroughly explored and answered, but we feel that the smart money is on complete chemical stasis, which will stay this aging process. Yeah.
How long do those white blood cells last? They talk about re-injecting them into people. I mean, but you aren't injecting stem cells. So once they are in there for a while, they'll just die and we'll go back to that situation.
Well, when we freeze the cells, we freeze a complete cocktail of the cells, including stem cells. And when they're thawed and infused into the organism again, they go through their normal mitotic division, and they proliferate. They are just as if they were 20 years old. Now, white blood cells last quite a long time. And with the stem cells, those youthful stem cell populations will recreate the immune system pretty satisfactorily. We have evidence that this happens. So I think we have a pretty sure bet that preservation of the immune system in liquid nitrogen over as much as 5 to 6 decades is quite feasible. Yeah.
I think we need to cut questions now. Ken and Chris have a table right outside the work.
Got a
They'd
table.
be happy to talk to you further.
Thanks for bringing